Hi, everyone. Welcome. We are going to get started very shortly. Welcome, everyone. Thank you for joining. We're going to get started in just a minute. Hi, everyone. Welcome in. All right, we are going to get started. So, welcome everyone to the PD Generation webinar for PD Generation at the World Parkinson Congress 2026. I'm so excited for you to be here with us today to talk about the PD Generation study and some really exciting discoveries within genetics and PD, and really just have a great conversation around genetics research. So, for today's agenda, like I said, we're going to start off by talking about the PD generation study, really what the study is, and hopefully spark your interest in research in Parkinson's disease. Then we're going to talk about how we're helping advancing research in the genetic space through GP2 or the Global Parkinson's Genetics Program. And then we're going to talk about how PD generation is coming to WPC in May of 2026 and how potentially you can get involved. We'll have some closing remarks and then a Q&A as well. We do have a Q&A section, so if any questions come up throughout this presentation, please feel free to stick it into the Q&A, and we'll have some time at the very end to address as many as we can. So, to get us started, I'd like to introduce myself. My name is Kamalini Ghosh Galvelis. I'm the Associate Vice President of Clinical Research at the Parkinson's Foundation, and I lead the PD generation effort. I'm really excited to talk to you about this research study that the Parkinson's Foundation started in 2019 and give you a perspective of what research could look like in the Parkinson's space. But before I get started, I do want to quickly touch upon what the Parkinson's Foundation is. We are a nonprofit, and our mission is to make life better for people living with Parkinson's disease by improving care and advancing research towards a cure. Everything that we do really involves the passion and the energy and the experience of the global Parkinson's community, and that includes our research. And so we really hope that everything we do can help make life better for people living with PD. In order to accomplish that mission, we have three goals that we like to keep in mind here at the Foundation. One, we want to improve care for everyone living with Parkinson's disease. We want to advance research towards a cure, and we want to empower and educate our global community. And we know that there is a big urgency in order to achieve these goals and the mission. We know that this urgency is because Parkinson's disease is the second most common neurological disease after Alzheimer's disease, and it impacts 10 million people worldwide and a million people living within the United States. There is quite a bit of an economic cost to Parkinson's disease just because there's no cure currently, and all of the treatments out there are really symptomatic based. It is the fastest growing neurological disease in the United States. And with that in mind, we are making sure that our mission is always a top of mind and we're doing everything that we can to really reach for that cure. So, diving into the research team at the Parkinson's Foundation, that's the team that I'm a part of, we also have three core pillars: basic science, virtual biotech, and PD generation, which is clinical research. So, today I'm going to focus in on PD generation. So, to really set the stage, the way that I like to think about this is like a fingerprint. And we know every person's journey with Parkinson's disease is unique, like a fingerprint, and it's made up of a lot of different elements. It's made up of the journey to the diagnosis, the clinicians that you see, the treatments that you take, your support system, like your friends and your family. And we also know that one element of this fingerprint is genetics. And we think genetics could serve as a key to understanding the biology of Parkinson's disease. It could help us understand better diagnostic tools and down the line help us find treatments for Parkinson's disease. So it sounds like genetics is pretty important. So you would think, oh, maybe a lot of people understand their genetic status in Parkinson's disease. But if you go back to 2019, you'll see that actually is not the case. So most people in 2019 did not receive genetic testing. And the reason for that is because it was pretty costly back then. And because there was no impact on care, there was a lack of insurance coverage for genetic testing and counseling. This made testing pretty inaccessible. There were a few direct-to-consumer panels back in the day, which consisted of pretty limited genes like LRRK2 and GBA1 and just a few variants within them. But most importantly, these direct-to-consumer panels were not accompanied by genetic counseling. And we think genetic counseling can really serve as a way to understanding how genetics impacts your Parkinson's disease fingerprint and journey. These two together created a major barrier for the success of genetic trials within the PD space. Because as you can imagine, as a clinician or potentially a person living with PD, if you don't know your genetic status, you might not know that you're eligible for genetic trials in the future. And that could also mean that there's a risk for different companies to actually fund genetic trials. All of this combined led the Parkinson's Foundation in 2019 to start PD Generation. PD Generation is a clinical research study that offers genetic testing and counseling to anyone that has a diagnosis of PD in English and in Spanish. So, what makes it unique? We kind of touched upon it a little bit, but we know that PD Generation offers a CLIA certified or a clinical certified genetic test to anyone who has a diagnosis of Parkinson's at no cost to them. And really, this is inclusive of anyone that has a diagnosis, no matter of age, location, race, or gender. We also offer genetic counseling through this study so that you not only get to learn your genetic status, but you also get to have a conversation with someone around the genetics and your specific genetic status so that you can better understand how genetics might play a part in your fingerprint and have better discussions in the future. So what actually is entailing in PD generation? What's involved as a participant? What do you have to do? So this is the general participant workflow, and you'll actually see this come up further down in our slides for WPC, but I'm going to walk you through basically the day in the life of a participant in PD generation. So we're going to start up here on the left-hand side with a consent, and this can happen either at your house or in a clinic that you're being seen at. So a consent is basically an opportunity for you to learn about what PD generation is, understand the risks, the benefits, and really make a decision of whether you'd like to participate in research. And so once a consent is done, then we collect a little bit of clinical data. So this clinical data allows us to understand a little bit more about who you are and a little bit more about, your clinical diagnosis, and helps us tailor a lot of the conversations for your genetic counseling session. From there, we do a blood collection. So the blood collection is used for us to do a whole genome sequence that's looking at your entire DNA and then really pinpointing and analyzing specific genes that we know relate to Parkinson's disease. So our lab will do all of that analysis, and then you'll be given your results back for this primary panel through a genetic counseling session. Once the genetic counseling session is completed, you'll get your results through the mail or email, and then you'll have the opportunity to participate in secondary health findings, so even more genetics results. And all of the data that we collect is actually given back in a de-identified manner to researchers and scientists, such as the ones at the Global Parkinson's Genetics Program, so that they have a library of information to really evaluate within the Parkinson's genetics space. So you might be asking, well, what genes are you looking at? We have three specific panels that we run at PD Generation. The first is the primary 7-gene panel, and that consists of genes like GBA1, LRRK2. These are genes that are classically evaluated within the Parkinson's space, and we know that they relate to Parkinson's disease. We also have two optional panels. So one 21 related Parkinson's panels. These are exploratory genes within the Parkinson's space. And then we also have 10 non-Parkinson's related genes. And these genes relate to other conditions that could have family implications, such as breast and ovarian cancer, Lynch syndrome, and then familial hypercholesterolemia. So this is a comprehensive panel for anyone that comes into the study, and you have the option of just having the 7 gene panel run or all of the panels run. And really the goal here is for us to give back more information so that you are just more knowledgeable about that genetic portion of your fingerprint. So, who can participate in PDGene? We have tried to make this study as accessible as possible. So there are some inclusion and exclusion criteria that I want to go over with you today, but we really do try to ensure that all people that have a diagnosis of PD have the opportunity to participate in research. So for the inclusion criteria, you have to have a confirmed diagnosis of PD through a clinician. You have to have the willingness to undergo genetic testing and then choose to be informed of those genetic testing results, have to have the capacity to give full informed consent or have a legally authorized representative consent on your behalf, and have the ability to perform all of the study activities. So again, we tried to keep it as inclusive as possible. In terms of exclusion, there are a few things that we have to keep in mind when it comes to evaluating a blood draw and blood samples when we're giving back results related to Parkinson's disease. So these are few exclusion criteria that we have. First, if you have a diagnosis of an atypical Parkinsonian disorder, if you've had a blood transfusion within the last 3 months, if you are currently having an active hematologic malignancy, such as leukemia or lymphoma, or treatment for those, or if you're under the age of 18, at this time we won't be able to enroll you into the study. So another question in research is, well, what are the benefits of participating in PDGene and what are the risks? So I wanted to walk through all of these with you so you can get a sense. So starting with the risks, anytime you participate in clinical research or give your information out to someone, there's always a potential for invasion of privacy or breach in confidentiality. And the way that we try to minimize this is de-identifying all of the data that goes external to researchers and scientists. And we do this by stripping of all the identifiers and giving a specific code called the PD gene ID to your sample. And this drastically minimizes risk, but it's always something to consider when giving your information out. We also know that there might be a chance that there is a lab error or human error and we might not get results back to you. And if that's the case, we'll always let you know that's what's happened. And you can always give an additional blood sample or blood draw or decide to withdraw from the study at any time point. And then the last risk is because we're looking at genetics, and we know that genetics connects you to your past as well as your future and to your family, there could be an increase in worry and anxiety over receiving genetic results, which is why we felt it was extremely important for us to include genetic counseling as a part of this study. So you'll be able to talk about all of your concerns and your questions with a genetic counselor to really ensure that you are getting all of that answered when you're getting your results back. And then, so those are the risks for the study. Some of the benefits are from the study are that you actually get to understand your own genetics for Parkinson's disease and really get to have better conversations with your family members, with your clinicians around what that can mean for you. But also, you're going to help accelerate Parkinson's disease research and move it forward by allowing researchers and scientists the opportunity to tap into this brand new de-identified library of genetic information. So now what I want to do is kind of give you an idea of who's participated in PDGene so far and where did they come from. So this is showing you our cumulative enrollment in PD Generation from 2019 to about 2 weeks ago, so mid-January of 2026. And this is the trajectory of the enrollment. So you can see here, scattered along the way, are a few milestones that we've reached. I think some of the ones that I want to point out are we enrolled 10,000 participants a year ahead of schedule, and then we also partnered with Aligning Science Across Parkinson's and the Global Parkinson's Genetics Program to really help get the word out, think about inclusivity and diversity and expanding upon the accessibility of this study, which was an amazing partnership that we've been able to continue to this day. And then we reached 30,000 participants in December of 2025, which was just something we were so excited and proud of, which means 30,000 individuals have been a part of the study, have contributed their data to science, and then have also received results around their genetic status in PD. So to give you more of a perspective of the enrollment for this study, in the middle of this slide is the enrollment per country. So PD Generation is operating in quite a few countries, which we're really excited about. And you can see the breakdown of individuals per country in the middle of this map. Also on this slide are a few more identifiers of our cohort. So the average age of individuals in our cohort tend to be around 69 years old of enrollment. We have about 58% of our cohort identifying as male, and 23% were diagnosed in the past 2 years. And really a number that I think is pretty astonishing and I love to point out are these two numbers up here. So 74% of our full cohort and 81% of our Hispanic cohort have never participated in research before. And I think that shows us two main things. The first thing is it shows that PD generation is an accessible study and is really open to individuals who might not have access to clinical research based off of where they're living. But we also know then that PD generation acts as a gateway to research for participants to get their hands wet in research, see how it feels to contribute, and then decide if they want to continue to participate in the future. So we're really excited that these numbers continue to increase and that this study is really backed up by the Parkinson's community. And so one of the main questions that we get is, oh, well, how many people with Parkinson's have a genetic component to the Parkinson's disease fingerprint? And so what we found through this study is that 12.1% of individuals in PD generation have a genetic component to their Parkinson's disease. So what you'll see here is a double pie chart where on the left-hand side, you'll see 12.1% of our population testing positive, and of those individuals testing positive, what they tested positive in. So majority test positive in GBA1 and LRRK2 and Parkin, but we also see here a few individuals testing positive in some of our more rare genes as well. And I think this gives us a really good understanding of the population of people living with PD that have a genetic component to them, but also gives us a lot more questioning of, what about everyone else and what's going on there? Are there exploratory genes that we need to look at? So it's exciting for us to have these numbers and be able to really move that forward. Also, one of the main questions we get in PD generation is, what is happening with my data? Is it just sitting around? Are people actually looking at it? So, like I said, in 2024, we started a formal partnership with Aligning Science Across Parkinson's Coalition, as well as the Global Parkinson's Genetics Program. And we have been able to contribute over 16,000 participants worth of data to scientists and researchers at GP2 in order to allow them to explore more genetic information within the PD space. So it's been really amazing to have this partnership and have individuals and scientists really look at the data that you're contributing. And with that, I would like to pass it over to my colleagues at the Global Parkinson's Genetics Program. It is my distinct pleasure to introduce our next two speakers, Dr. Andy Singleton and Dr. Alistair Noyce. So Dr. Andy Singleton is a world-renowned geneticist by trade, and he has focused his career on genetic foundations of neurodegeneration disease, and more recently on the genetic foundations of Parkinson's disease. Notably, his lab identified LRRK2 mutations as a novel cause of disease in Parkinson's, and he is the co-lead of the Global Parkinson's Genetics Program, which again is an ambitious initiative of the Aligning Science Across Parkinson's Coalition. We also have on the call today Dr. Noyce, who is an amazing neurologist and professor of neurology and neuroepidemiology at Queen Mary University of London. His research group focuses on environmental, clinical, and genetic factors of PD, and he plays an important role in the Global Parkinson's Genetics Program as well. So I'm really excited for Dr. Singleton and Dr. Noyce to be here with you today to talk about how GP2 is contributing to the bigger picture of genetics and Parkinson's disease, the data that's coming out of GP2, as well as how PD generation helps contribute to that picture. So I am going to pass it off to Dr. Singleton and Dr. Noyce. Thank you both for being with us here today.
Thank you very much, Camerlini, for that very warm introduction. Do you want to say hello as well, Andy?
Yeah, hi everybody. Thanks for joining today. Alice is going to lead us through this. Please, if you have questions, just put them in the Q&A or save them for the end, and we'll try to answer as best we can.
Thanks, Andy. So following on from Camerlini, I guess the question is, why does GP2 support PD gene, and why does PD gene share data with GP2. And so I'm going to walk you through some aspects of the Global Parkinson's Genetics Program and some of the key things that we're working on right now. So next slide, please. So the aim of GP2, the Global Parkinson's Genetics Program, is to dramatically expand our understanding of the genetic basis of Parkinson's disease and related disorders and make that knowledge globally relevant. This is a 10-year program that began right at the beginning of 2020 and that is funded through to the end of 2029 with this mission at its core. Next slide, please. So why do we want to do this? Well, we want to understand the genetics of Parkinson's because genetics tells us about the disease at both the molecular and cellular level. And that means what changes are occurring to initiate the disease and then also drive the disease forward. If we understand these changes, then we can aim to interrupt or reverse the disease process, not just by treating symptoms, which is what we can currently do for Parkinson's pretty effectively, but with a comprehensive understanding of these changes, we can aim to stop, slow, or reverse the disease. And in time, we hope to match drugs to the molecular events that are driving disease in an individual. This is the concept of precision medicine, matching drugs that are targeted against particular genetic defects that occur at the individual level, precision medicine. And this is a concept across other disease areas such as cancer therapeutics. But another major aim of GP2 is to capture, as Camelini's already touched on, an understanding of the genetics in people who haven't previously been involved in research. And Parkinson's is a global disease. If we want to treat everyone, we need to understand the disease process in everyone. And so there's this nice expression that a rising tide floats all boats. If we understand how the disease is different and the same across populations, then it will accelerate discovery for all. Next slide, please. So what we're aiming to do in GP2 and in genetics research in general is identify a part of the genome that is linked to disease. Within that part of the genome, hone in on the gene that is driving the disease, the causal gene, start to understand what the functions of that gene are, and within that understanding, identify targets that can be modified with drugs. By understanding the biology that exists within a disease, we can start to dissect it, all the different pathways that are contributing, and then develop drugs to slow down or stop the disease. And there are some really nice examples of this already. Next slide, please. So some of the genes that you'll hear about, and some of the genes that have already been mentioned by Camelini and will come up later in these slides, are the genes for the gene for alpha-synuclein, the gene for GBA1, glucocerebrosidase, and the gene which is called leucine-rich repeat kinase 2, or LRRK2. And Andy Singleton, who's joining us on the call and who'd be happy to take questions on these, really played a fundamental role in identifying alpha-synuclein and LRRK2 in particular as causes of Parkinson's. Now, this slide's quite text heavy, but there are mutations in all of these genes that are known to cause Parkinson's disease or substantially increase the risk of Parkinson's in people that carry them. And over on the right here, you can see a figure which is a summary of all the drug trials that are ongoing in Parkinson's right now. In the top half of the figure, it's the drug trials that are aiming to modify the underlying disease process. In the bottom half of the circle, it's the drug trials that are aiming to improve symptoms. And within that figure, I'm not sure how well it projects, but you can see in the top half the compounds that are shaded in purple are those that are targeting GBA. The compounds that are shaded in green are those that are targeting alpha-synuclein, and the ones that are shaded in yellow are the ones that are targeting LRRK2. And for all of these different causes of disease, there are actively recruiting drug trials going on right now. Phase 3 drug trial aiming to clear alpha-synuclein, taking place in many countries around the world. A couple of large trials taking place to increase glucocerebrosidase activity, and then 2 trials at phase 2 aiming to reduce the activity of LRRK2. So you can see that the discoveries made from genetics down the line directly inform the design of clinical trials. And so next slide, please. So what's GP2 aiming to do? Well, there are really kind of four main pillars of GP2. That is developing global collaboration and capacity building. Okay, we need partners all around the world to contribute to GP2, to generate data, to collect samples from patients and non-patients, and to support them in setting up research infrastructure. Just down from there is growing the next generation of leaders. And so GP2 is absolutely committed to instilling skills and knowledge in early career investigators around the world, so that once you've built that capacity in different parts of the world, there is an influx of young talent to occupy places and drive the field forwards. In the top right corner, we use transformative genetic methods, the cutting edge, state of the art methods to dissect the disease and make the most of the data that is generated by GP2. And then again, fundamentally important in the bottom right of this figure, making sure that the data is available to all. This is not a program that aims to harvest data and samples from countries all around the world and then use it for writing research publications for a few investigators. We're really investing in global infrastructure to allow investigators all around the world to answer the questions that they have using the data that they and the wider GP2 community is generating. And so all of that together creates this self-propagating discovery engine to make transformative discoveries. And we're going to talk about some of those discoveries in the next few slides. Next slide, please. Okay, so I talked about training, and there really is a kind of smorgasbord of training opportunities for people all around the world. We have developed an enormous amount of online content to maximize training opportunities to a wide number of people. We set up a trainee network which has more than 250 members around the world. And those trainees that are really leaning in hard to the trainee network and consuming the online information and training that's available then get the opportunity for further training opportunities. So training placements at well-established labs, the opportunity to undertake masters projects in Parkinson's genetics, to undertake PhDs, to go to workshops around the world to really develop their skills, and the layering knowledge on those trainees so that they can make their own discoveries driving forwards. Next slide, please. This is just a summary of where you can find the GP2 trainees. So again, there are plenty of trainees in North America and Europe, but I think the most exciting thing here is that we have, nearly 80 trainees in Latin America, 90 across Europe, nearly 70 in Africa, 10 across Oceania, and all of this contributing to more than 250 members actively engaged in the training and networking group. Next slide, please. GP2, when it started out, was really its focus was to identify where data had already been collected, genetic data and clinical data, and how that could be brought together from all around the world into a data resource for the research community. But over the last few years, GP2 has been able to support lots of new collections across all 6 continents. And I won't go through these one by one, but you can see that we really have a very broad scope in where we've supported further collections for tens of thousands of samples. Next slide, please. And when we set out, the aim was to generate data on more than 100,000 participants by the end of GP2. We've already released data from more than 100,000 participants in the latest release, which was in December 2025. And as Camerlini said, a large number of those samples come from PD gene participants. So when you ask the question, how is my data being used? This is how it's being used. It's going into a central resource along with data from people all around the world that are contributing to research to make these transformative discoveries. And now our ambition has grown much, much more. Now that we're supporting data collection in lots of different places, PD generation included, we expect to have more than 340,000 samples released by the end of 2029. Next slide, please. So this was the question, does my data really get used for research? And as I've said, we've created this resource that is now being used by thousands of researchers all around the world to ask critical questions. We make the data available, we provide the computational power to do the analysis, and we train the next generation of scientists to do this. And this was what makes GP2 the discovery engine. And to labour this point about it not the project's not just being led by a handful of researchers in North America and Europe. There are now 150 discrete research projects that have been submitted by the wider GP2 investigator pool, and that really shows how much the resource is being used. And so here we'll just go through some of the highlights right now. Okay, next slide. So I think this is a very nice example. I don't think this has been published just yet, Andy, but it's at a very advanced stage with a journal, with a very large journal. And this is really a summary of the global genetic spectrum of Parkinson's disease, broken down by different genetic ancestral groups. So EUR stands for European, AFR stands for African, MDE stands for Middle East, CAS, Central Asian, EAS is East Asian, and SAS, South Asian, AJ, Ashkenazi Jewish. And so these are all different ancestral groups. And this is using data just from people with Parkinson's. So there's no control data here. Just in people with Parkinson's, you can see the distribution of the different type of gene mutations and variants that exist. Okay, so if you look right at the center of the map, there's the European ancestral group, EUR. You can see that most of the variant, more than about 3,000 samples in that. Most of the gene carriers are GBA1 carriers. We heard Camerlini mention GBA1 already, but there's a good number of LRRK2 carriers within that group. I think it's interesting if you look across Asia, so the Middle East, MDE, Central Asia, and East Asia, you can see how the distribution of different genetic variants varies across Asia, which I think is really interesting. And then if you look with where the pins are located, you can see where there are trials already set up that are focused on GBA1, on LRRK2, and some of the recessive forms of Parkinson's, Parkin and PINK1. And you can see that most of the concentration of these trials is in Europe and North America. This is something that we aim to change as we further expand our understanding of the genetic base of Parkinson's. We expect that these pins will start to be distributed much more widely around the globe. Okay, next slide, please. Now, this is a complicated slide full of lots of numbers and full of lots of text, but whereas the previous slide showed in cases only, in people with Parkinson's only, the different types of mutations and variants that they carry, the next set of slides is all about an approach called genome-wide association studies. And what genome-wide association studies aim to do is take thousands of people with a disease like Parkinson's and compare them to thousands of people without the disease. We call those healthy controls. And you can see the numbers at the bottom here. In this European GWAS, there's 63,500 cases and more than 1.7 million controls. And what a genome-wide association study does is it looks across the entire genome, all 22 chromosomes, and looks for very small differences in particular variants that exist within genes and within loci. And by doing that, you can get a broad overview of all the different genetic contributions that underpin sporadic Parkinson's, i.e. non-familial Parkinson's. And the next slide is an informative slide for how that looks. So we call this is the output from a genome-wide association study. Along the bottom, you can see all the chromosomes numbered there, and they start with chromosome 1 on the left, that's the largest chromosome, goes through to chromosome 23, that's the X chromosome, so 22, smallest chromosome. You can see they get smaller as you go across. The x-axis, the height of these peaks, is how significant these different genome-wide association study hits are. Okay, and the height of that peak is really an estimate of how unlikely this peak is due to chance. And so the higher the peak, the less likely this is a chance finding, the more likely this peak has something to do with Parkinson's disease. And so if you look at the top peaks, we call this a Manhattan plot because it looks like the Manhattan skyline. And you can see as you look across the top from chromosome 1, you see that GBA1 variant there. So this is a cause of sporadic Parkinson's, or we think that the GBA1 pathway is important in sporadic Parkinson's. As we go across over chromosome 2, you can see SNCA, which is the synuclein variant there, and as you go across to chromosome 12, LRRK2. So we know that mutations in each of those genes can cause familial Parkinson's. By doing the genome-wide association study, we also know that those genes are likely to be important to seemingly sporadic Parkinson's or non-familial Parkinson's, which increases our confidence as we're starting to do drug trials for all of those three targets. So that's the European Ancestry Genome-Wide Association study. It's the largest one so far. Next slide, please. Now, this is really interesting. I think for me, this is my favorite output of GP2 so far. But I need to tell you, and I'll show you on a subsequent slide, that the information, the number of hits we get from a genome-wide association study increases as a function of the sample size. So the more cases, the more healthy controls go in, the more hits you tend to get. This is a relatively modest genome-wide association study undertaken in West African patients and healthy controls. I think it had, I forget off the top of my head, was it about 1,000 cases, I think? 1,000 or just over 1,000 cases, Andy?
Yeah, it's about 1,500 in total.
1,500. And then about 200,000 healthy controls. So this is pretty small as a genome-wide association study goes, but there was a single hit, and it's a very strong hit. You can see that peak in chromosome 1, and the variant that it tags is also a GBA1 variant. So what does this tell us? It tells us that in a relatively small genome-wide association study, there's a dominant hit, and it's in a known Parkinson's gene. And not only is it a known Parkinson's gene, but this particular variant in the GBA1 locus is present in about 50% of African patients with Parkinson's disease. In about 40% of West African patients with Parkinson's, they have one abnormal copy of the variant. In about 10%, there are two abnormal copies. And this is really interesting because that suggests that GBA1 is a dominant mechanism in West African patients with Parkinson's. And then again, this opens up the opportunity for precision therapeutics and targeted drug trials. So I think this is really, really important. And some of the follow-on work that was led by Angelica Okubadejo and Cornelis Blauendrat has really started to unpick what that variant is doing and how it may be causing Parkinson's disease. Next slide, please. I won't dwell on this too much, but this is another very nice example of how adding in data from diverse ancestral groups really starts to increase our knowledge. So this genome-wide association study included mainly European cases and controls, but also some East Asian, Latin, and African cases, relatively modest numbers compared to the European case control data set. But what it identified was 12 new loci that didn't come out of the European GWAS so far. So just by putting in small numbers, relatively small numbers of East Asian, Latin, and African patients and controls, we start to see new signals which start to tell us about what may be driving Parkinson's across as a global disease rather than just as a European disease. Next slide, please. And so I think this is a useful figure. And so you can see that this is the bottom axis here is time, and the upright axis, the y-axis, is the number of hits you get from a genome-wide association study. And so you can see back in 2006, when there was the first Parkinson's disease GWAS with only 200 cases, 200 controls, there were no hits. Then the next Parkinson's disease GWAS had 2,000 cases, 18,000 controls, and there were 3 hits. By 2011, there were 16 hits, 2014, 28, and so on and so on. Now we're in 2025. The latest European Ancestry Parkinson's GWAS has 134 hits. So you can see that point I made earlier on, that as you increase the sample size, you start to understand more about the drivers of disease and find more loci. And so that's just in red. But if you look in blue, that's the East Asian genome-wide association studies. Now, increasing number of hits from genome-wide association studies. In orange, the Latin American. In purple, the South Asian. And I think the first and now second African genome-wide association studies. And again, as we get to 2025, some of the hits coming out of the African Genome-Wide Association are some of the best established hits in the European Genome-Wide Association study as well. So this tells you that, shooting for 350,000 participants in GP2 is a really worthwhile exercise. We will find out more, better understanding of the drivers of disease, and better opportunities for precision medicine going forwards. Next slide, please. So what next? Well, we're in 2026, 6 years into GP2, and I think we feel that GP2 is just hitting its stride. There is an awful lot more discovery and faster analyses planned over the next period. We've got tens and tens of analyses planned, even over the next 12 to 18 months. But back to this question about how your data is being used. Your data is being worked on by trusted researchers all over the world to answer critical questions about Parkinson's disease cause and progression. Okay, what causes the disease? What influences progression? And is this the same across populations or across individuals? And I hope bringing this back to PD generation, you see PD generation as an opportunity for you to contribute to this effort. So with that, I'll pass back to Camerlini and to talk about what's planned for PD generation of the WPC. You can skip over this one.
Okay. Thank you so much. This every time I hear this presentation and I hear more of what GP2 is doing, it is just so wonderful to see that so many people are looking at this and really focusing on not just Parkinson's disease, but genetics in Parkinson's disease. And I really see the vision. And thank you again so much for explaining it all to us and kind of showing how PD generation contributes to this. So now what I'm going to do is I'm going to bring it back to PD Gene and kind of explain what we're going to be doing at WPC this year. So, this is going to be one of the first times that PD Generation is brought to a conference stage in this manner. So, we're going to be doing in-person live genetic testing and counseling during WPC so that anyone who has a diagnosis of Parkinson's disease at the conference can participate in research in real time. We'll have a lot of researchers and scientists from GP2 there as well. So, you'll be able to chat with a lot of different people that are not just doing the clinical research side of things, but also really looking at the data and trying to move the needle forward. So we're going to be testing from basically all the days of the conference from May 24th from 7 to 9 p.m., as well as May 25th to the 27th, the entire time the conference is running. And really, this opportunity is not just for individuals who have never heard of PD generation or have never contributed to PD gene before, but also we're going to be having some new research opportunities for individuals who have already participated in PD gene. So whether you're new or you've already done PD gene, there's going to be an opportunity for you to participate and continue to participate in research if you would like to. Like I said, the research team at the Parkinson's Foundation as well as GP2 will be there, so you can always get your questions answered. So I wanted to bring it back to this flowchart, right? So what exactly is going to happen at WPC? So we talked about the different elements of PD generation, and at WPC, we're going to start with the first three elements. So the consent in English or Spanish, the clinical data collection, and then that blood draw. And then from there, all of that will get processed at our lab, and then we'll move forward with your genetic counseling and so on and so forth. So again, this is an opportunity to participate in research, to contribute to research so that we can continuously understand the genetic basis of Parkinson's disease, and really get a chance to talk about this really amazing project in person. And like I said, if you've done PD generation already, there will be opportunities to participate in other aspects of PD gene as well. This year, we've expanded to 3 different sub-studies. They include PD gene surveys, PD gene family, and PD gene insights. I'm going to quickly walk you through what these three new sub-studies are. So PDGene surveys is a way for us to poll the entire PD generation cohort around topics of interest from researchers and scientists, such as the ones at GP2. So we ask about environmental exposures, potentially diagnosis changes, medication changes, interest in clinical trials, and things like that. And again, that is for our chance to basically get a pulse from the PD generation cohort and really share that back with researchers and scientists. There also may be some opportunity at the WPC to get your family members involved. So if you have biological family members who are coming to the WPC, they may have the opportunity to also get genetic testing and counseling done as well. And then the final opportunity is going to be PDGene Insights. So our goal, now that we have a lot of genetic information, is to supplement that genetic information with more clinical data. We want to understand your journey to the diagnosis. We want to understand what symptoms you're experiencing. We maybe want to see if you have loss of smell. So there are different ways for us to really gather more information to supplement your genetics and really share that back with the researchers. And that's going to be through PDGene Insights. So again, these are more opportunities to participate if you've already done PDGene. So now the main question is, well, how do I participate? How do I get involved? I think step one, which I'm sure many of you have already done, is to meet us at WPC. So register for WPC and really get involved through actually being at the conference. But there's also other ways to interact prior to actually meeting us at the conference. And so you'll see a QR code on the right-hand side of the screen. That QR code is a survey that the PD Generation team has generated for us to get to know you a little bit more. So we want to understand, a little bit more about your Parkinson's diagnosis. We want to understand if you've done PD Generation and what other opportunities you might be interested in, such as all of the sub-studies. So if you want, you can scan that QR code there, fill out the survey, and we'll be in contact with you shortly to do some pre-consenting and really get an understanding of how you can contribute to research if you so choose to do. And then step three, once those steps are done, come meet us at the PD Generation booth and we'll do your sample collection. And so we're really excited to offer this opportunity at WPC. Like I said, it's the first time PD Generation is mobilizing to this level to really bring research to the community. And so we're very excited to see you there. And in case you have more questions or you want to think about it more and get more understanding of what actually is happening, there's a few different resources here. So you can always check out our Parkinson's disease PD Generation website. You can join another webinar. We have two more webinars. We'll talk about very similar things here. One that's going to be in Spanish on March 25th, and then another one in English that's going to be on April 15th. Again, for a chance for you to hear the same presentation, but also reach out and ask your questions. And the last stage, of course, is you can register for WPC and learn more on the ground in person about PD generation, opportunities to participate in research, and really about what the Global Parkinson's Genetics Program is doing to advance Parkinson's care and research. And with that, I would like to thank you all for coming and for listening to our presentations. I also like to thank our two speakers, Dr. Singleton and Dr. Noyce, for giving us an amazing overview of what GP2 does and how it contributes to advancing research in Parkinson's. And now what I want to do is I want to open it up to questions that you might have as potential participants or just individuals coming to learn more about the study. So I'm going to actually ask my colleagues to come on and see if there's any questions that we'd want to answer.
Yeah, thank you to all of our speakers for amazing presentations today. We have some questions that have come in through the chat. The first one is for Dr. Noyce and Dr. Singleton. I know you talked about this a little bit, but can you just summarize why is it so important for more people to join PD Generation and contribute their data to science?
Yeah, maybe I can answer that, and Alistair, you can correct anything that I say that's wrong. So one of the key things I think to understand about the work that we're doing is that the work that we're doing is it's not just about finding genetics and finding genetic causes and risk factors for disease just to do that. It is all geared towards finding treatments for disease. For every gene we find, every new risk factor we find, that is a new shot on goal. It's a new potential therapeutic target. It tells us more about mechanisms that underlie disease. It tells us about which populations we should start to prioritize for therapeutic trials. And it really reduces it reduces the activation energy that trialists need to kind of move forward and create new therapies and test new therapies. The power for detecting new loci is and new risk factors is as Alistair mentioned, linked to numbers. So the more participants that get involved, the more clinical information they provide, the more power we have, the more things we'll detect, the more chances we have to find new therapeutic avenues. And as you can see, to do these kinds of studies, we need hundreds of thousands of individuals, and every single person counts. Um, every person that takes part increases the number of shots we get on goal, increases our chances of finding something new and novel that will point us towards the therapies that are being developed.
Great. Thank you so much for that summary. One more question for Dr. Noyce and Dr. Singleton is, why is diversity so important in genetic studies? Not only racial and ethnic diversity, but what about also like geographic diversity?
Yeah, well, I can take this. I mean, I think what is true, not just for Parkinson's, but across complex disease of which Parkinson's is one, is that 90% of the data that's been generated so far comes from 15% of the world's global population. That's the European population. So there's this huge mismatch between the data that we have at our fingertips and our understanding about the basis of disease and who is actually affected by those diseases. And so that is a huge gap that GP2 and its focus on Parkinson's is trying to address. Now we can um, we can just focus on diverse patient and healthy control groups within countries like the US and Europe. But what we really want to do is make this less Eurocentric, less US centric, and really, think about those four pillars that I pointed out earlier on, really start and drive capacity building across the world. So understand about the cause of disease in different diverse ancestral groups, but also build capacity because as Andy said, all of this is not just about finding new genes and publishing research papers, but about creating actionable targets for drug trials and new therapies. And it's a global disease, and that only serves a purpose if ultimately we arrive with new drugs that are spread and delivered across the world.
Yeah, just to add to that, Alistair's completely right. I think we can think of these things in two ways, really, right? There's the social imperative. Parkinson's is a global disease. We should be aiming to treat disease in everybody, right? Equity is important. There is another side to it as well, which is there's a scientific imperative too, which is that by looking across populations and looking at what's different, what's the same, it gives us incredible scientific insight that we just would never get by looking at one population. So Alistair showed the example of finding the GBA risk factor in West Africa. We learned so much about GBA from looking in that population. It was a new mechanism, changed age of onset, a whole host of new things. And we just never would have found those things out if we'd just been focusing on one population. So it really is this, it might sound trite, but it is this idea of a rising tide floats all boats. The more we learn about each population, the more we learn about all populations.
Great. Thank you both so much for highlighting some of the gaps and why it's so important for us to consider a global perspective. Kamalini, next question is for you. What if I do not know or remember if I participated in PD generation before? What should I do or how should I proceed?
Sure, that's a great question. So I think the best way to proceed is to let the PD Generation team know who you are, and we can try to make sure we get you your data or information, or help you sign up to participate in PDGene if you want to. I think the best way to do that is, let me go back two slides, is this QR code. So this will give us the opportunity at PD Generation to get to know you a little better and identify whether you've done PDGene in the past. And if you have, we would be happy to reshare your genetic report and your information. But if you haven't, we'll help you get signed up for WPC if you're going, or just through the regular PD Generation pipeline. So yeah.
Great, thank you. And then another question for you, Kamalini. How do I spread the word about PD generation at the WPC locally? Do you have flyers I can share? What is the best way?
Oh, that's an awesome question. Okay, thank you. Yeah, definitely. We have a lot of information that we'd love to share with you. If you want to email us, let me go back to that email. There we go. You can always email us and we can send you over a couple different flyers that we have around testing at WPC. We can send you over virtual links, such as the one at the WPC website, but we'd be happy to share a lot of flyer and information with you to spread the word. Thank you for doing that.
Great. Thank you so much. And then one final question for Dr. Noyce and Dr. Singleton. I see a lot of your research involves people with Parkinson's. What are your thoughts on involving people who are also maybe at risk for developing Parkinson's? How are we including them in research and why is it so important?
I can take that first if you like, and then Andy jump in. So I think it's worth thinking about what it means to be at risk of Parkinson's. And so, from a genetic point of view, carrying a change in a gene which is known to be sufficient to cause Parkinson's in the future is one way that you might be at risk of Parkinson's. That affects a relatively small number of people. Um, then there are gene variants which increase your risk of Parkinson's, but in no by no means mean that you're predetermined to ultimately get Parkinson's disease. And you could extend that concept to look at all of those different variants I showed you in the Manhattan plot. From all of those different variants, we can build what's called a polygenic risk score, which is a composite score of how much genetic risk you have. And so we through GP2, we are recruiting people that carry variants in PD genes and have high and low polygenic risk scores. But then there's other risk groups as well. People that have unexplained smell loss, for example, people that have a certain type of sleep disorder with dream enactment, people that have significant problems with the automatic or autonomic nervous system. These are all things that are known to happen to some people before they get a Parkinson's disease diagnosis. And over the last couple of years, GP2's really expanded its focus on these groups. We focus on Parkinson's disease, other Parkinsonian disorders like PSP and multiple system atrophy, but also these at-risk groups, the people with dream enactment, behavior and smell loss. And so this is a focus because, one point of view is that at the point that someone gets a Parkinson's diagnosis, the disease has been there for quite a long time. If we only include those people, then we only learn about the genetic determinants of people that get that far. We want to know about the genetic determinants of the very earliest stages and also understand the genetic determinants of why people go from that early stage to a point of diagnosis. And that's why it's so important to include those people. And we're working hard on projects to try and understand how genetics drives that early progression from an early symptom through to the point of diagnosis. So it's definitely within scope, and we're very interested in those participants as well.
Great. Thank you so much. That concludes the questions that have been submitted. So.
Okay, amazing. Well, thank you again to Dr. Singleton and Dr. Noyce for being here with us today. And thank you all for joining and wanting to hear more about PD generation and how to contribute to research. Again, if you have any questions, feel free to email us to get in touch with the World Parkinson's Congress. And then hopefully we will see you there in person. So thank you again for joining, and we will see you in May. Thank you, everyone.
Thank you all.
Bye.
Bye.
Thank you.